The following recorded review will go over the study guide for Structure and Function Exam 2, over Chapters 7, 8, and 16. Chapter 7. Know examples of each type of bone and where you would find them: the long bone, short bone, irregular bone, and flat bone. So those are each of the types of 4 bones. You can see this on page 120, and they also give examples on page 120. So an example of the long bone would be the humerus or the arm bone. An example of a short bone would be the carpals or wrist bones. An example of a flat bone would be the frontal or skull bone. And an example of an irregular bone are vertebrae or spinal bones. It's important to mention as well that while some scientists recognize sesamoid or round bones as bones, this is not widely accepted. Therefore, there are only 4 official types of bones: the long bone, short bone, irregular bone, and flat bone. Next, let's look at the key terms: epiphyses, endosteum, diaphyses, and periosteum. Well, we can see all of these on page 120 as well as on page 121. So we know that the diaphysis is also the shaft. This is a hollow tube made of hard, compact bone, hence providing a rigid and strong structure, but also light enough to permit easy movement. The epiphyses are the ends of long bone, containing red bone marrow, which fills in the spaces of this spongy bone inside the epiphyses. The periosteum is a strong membrane of dense, fibrous connective tissue covering a long bone everywhere except for at the joint surfaces. At the joint surfaces, it is covered by articular cartilage. Endosteum is a thin membrane lining the medullary cavity. As you go through these and as you look at the image on page 121, it's important to note the layers that it goes in. Note that the periosteum is the outermost layer, followed by the endosteum, and then followed by the medullary cavity. Osteoclasts versus osteoblasts versus chondrocytes, their function, purpose, and location. We can see this on page 122 and 123. So we know that cartilage cells are known as chondrocytes, and they are located in the lacunae. We know that they help to build cartilage. So that is their function, purpose, and location. On page 123, we can see more about the osteoblast and osteoclast. They perform a process of constantly remodeling a growing bone as it changes from a small cartilage model to the characteristic shape and proportion of our adult bones, requiring continuous activity by osteoblasts, which are our bone-forming cells, and osteoclasts, our bone-reabsorbing cells. We know that the osteoblasts and osteoclasts are located Our osteoblasts are located in the lamellae of hard bone matrix, and when they get there, they become trapped and stop forming bone and are then known as osteocytes. Osteoclasts will release acids that dissolve the calcium crystals. This has 2 effects: removing the hard bone matrix and also allowing calcium ions to be released from the bone tissue, diffusing into our bloodstream and raising blood calcium levels. Appendicular skeleton. What is not included? So let's look on page 124 at our axial skeleton versus the appendicular skeleton. Now you can read about this on 124, and you can see a great image of this on 126. So we know that the bones of our center or axis of the body make up the axial skeleton. This will include the bones of the skull, spine, and chest, as well as the hyoid bone in the neck. The bones of our appendicular skeleton consist of all of the bones of our upper and lower extremities. True ribs versus floating ribs. What's the difference and which ones are which? Well, we can see more about this on page 131 going into page 132. So we know that we have 12 pairs of ribs that are all attached posteriorly to a thoracic vertebra, except for the lower two pairs, which are attached to the sternum and therefore have anterior and posterior anchors. Our first 7 pairs of ribs are referred to as the true ribs because they are attached to the sternum by costal cartilage. The remaining ribs are called false ribs because they don't attach directly to the sternum. You can see a great image of this also on page 132. Our false rib pairs are 8, 9, and 10, which attach to the cartilages of rib pair 7. Our last 2 pairs of false ribs are not attached to any costal cartilage, but appear to float freely in front, hence their name, floating ribs. Understand the vertebral column. So what order does it go in from superior to inferior? What is the largest section and what types of bones are these? So we can see a great image of this on page 130, and we can read about it on page 129. So we know that the vertebral column consists of a series of 24 separate bones, also known as vertebrae. We know that these are irregular types of bones. There are different sections of the vertebral column known as the cervical region, the thoracic region, and the lumbar region. There are 26 bones within the vertebral column. So it's important to note how the regions go of the spine in order of superior to inferior. So we know that superior is going to be to the top, so towards the head, and inferior towards the feet. So looking at the image on page 130, you can see that the most superior part of the spine or the vertebral column is the cervical vertebrae, followed by the thoracic vertebrae, followed by the lumbar vertebrae, followed by the sacrum, and finally ending in the coccyx. We can see that the largest section of vertebrae is the thoracic vertebrae, containing 12 vertebral bones. Know the major bones in the body, their names, and where they are located. For example, the bones in the forearm, the bones in the lower leg, major bones in the face, hands, and wrist. So looking on page 133, we can see that the bones of the forearm are the ulna and the radius, and the bones of the upper arm are the humerus. We know that the wrist and hands have more bones in them for their size than any other part in the body. We have 8 carpal or wrist bones and 5 metacarpal bones forming the structure or support for the palm of our hand. Our phalanges are finger bones. They are also our toe bones. On page 134, we can see the image of the hand bones. So we can see the radius and the ulna from the forearm connecting to the hand, our different carpal bones going into the metacarpals and then into the phalanges. On page 135, we can see that the bones of the lower leg are the fibula and tibia. We can see that the bone of the upper leg is the femur. We also know that toe bones have the same name as finger bones, phalanges. Our foot bones are comparable to the metacarpals and carpals of the hand, but they just have slightly different names. They're instead called metatarsals and tarsals of the foot. Just as each hand contains 5 metacarpal bones, each foot contains 5 metatarsal bones. However, the foot has only 7 tarsal bones in contrast to the hand's 8 carpals. The largest tarsal bone is the calcaneus or the heel bone. Trabeculae, definition, function, and location. So we'll flip back to page 121. So spongy bone contains many spaces, like a bath sponge. These cavities are filled with red or yellow bone marrow, and the beams that form the lattice of spongy bone are known as trabeculae. Fontanelles, what are they, when do they go away, and what purpose do they serve? We can see this on page 127. Fontanelles are soft spots on a baby's skull. These soft areas are 6 fontanelles, or areas where intermembraneous ossification remains incomplete at birth. They allow some compression of the skull during birth without much risk of breaking the skull bones. The fontanelles will fuse to form sutures before a baby is 2 years old. Oops. Sorry about that. We're still on chapter 7 though. I pressed the wrong button and I can't go back. But we're going to stay on chapter 7 for a few more things. So that covered fontanelles. And next we're going to look at examples of diarthrotic joints, synarthrotic, and amphiarthrotic joints. So we can see this on page 139. So we know that a synarthrosis is a joint in which no significant movement occurs. This functional characteristic is produced by fibrous connective tissue between the articulating or joining bones, which holds them tightly together. The joints between cranial bones are synarthroses and are commonly called sutures. An amphiarthrosis is a joint which only allows slight movement. Made up of cartilage, these join the bones tightly, but with a slight flexibility. Examples of this would be the symphysis pubis, the joint between our 2 pubic bones, and another example are vertebral discs, connecting each vertebral body to the next in the spinal column. Diarthroses allow considerable movement, sometimes in many different directions and sometimes in only 1 or 2 directions. We consider them to be freely movable joints. We know that we have several types of diarthrotic joints. Our ball and socket joint, which can be commonly found within the shoulder and hip joints. Our hinge joints, which can be commonly found in the elbow and knee joints. Pivot joints, which are commonly found in the atlas and axis of the skull, allowing the vertebral column the head to rotate on the vertebral column. We have saddle joints. A great example would be the metacarpal bone of each thumb and the carpal bones of the wrist. We have gliding joints. A great example would be the articulating processes in between the vertebrae. And we have condyloid joints, and a great example is the distal end of the radius into depressions in the carpal bones. Understand differences between the male versus female skeleton composition. So the main difference that we talked about in the difference between the male and female skeleton was that pubic the contrast of the male pelvis versus the female pelvis. So we know that male skeletons will have bones that are larger with more distinct bumps and other markings than female skeletons. The most obvious structural difference is in the pelvic girdle or pelvis. The female pelvis has a wide structure, allowing the body of a fetus to be cradled in it prior to birth, as well as a wide opening, allowing a baby to pass through it during birth. Whereas the male pelvis, coxal bones will form a more narrow structure than the female coxal bones do. The male pelvis is shaped more like a funnel than the broad, shallow basin of the female pelvis. Understand changes in bone density when we age. So we can see this on page 138. So we know that from the human skeleton is considered to reach its mature state around age 25. From then until about age 50 or so, our skeleton is in a state of active maintenance, continuously remodeling, dissolving, and rebuilding the bone tissue. But after age 50, the density of bone often decreases slowly due to a shift in remodeling activity. So bone density will decrease after age 50. Next, we'll look at the functions of the skeletal system, flipping back to the beginning of the chapter. On page 120, we know that our skeleton provides the internal framework of the body, like tent poles maintain the structure of the tent. Therefore, they provide support. Our skeletal system protects soft tissues located inside bony cavities. Our skeletal tissue, with the conjoined use of our muscular system, allows for movement. Movable joints within our skeleton make movement possible. Bones also play an important role in maintaining the homeostasis of blood calcium, a vital substance required for normal nerve and muscle function. They serve as a safety deposit box for this calcium, allowing blood calcium to move out of the blood into the bones for storage, and also allowing calcium to move in the opposite direction and enter the bloodstream, raising blood calcium levels. Our skeletal system also helps in blood cell formation, carried on in the red bone marrow, a process known as hematopoiesis. Articular cartilage we briefly touched on, and it's on page 120. You can also see a picture of it on page 121. So we know that our articular cartilage will cover the ends of bones, and it allows for that free movement so that the bones don't scrape against each other. And we also talked about the parts of a bone in chronological order from superficial to deep, which you can see on page 121. It would also be important to pay attention to examples of the movement of the types of movement that we can achieve with our skeletal system in conjunction, of course, with our muscular system. And these are going to be seen on page 142. An example of extension would be to extend the joint, for example, straightening a bent elbow. An example of abduction would be to abduct a joint, for example, moving the arm to the side and away from the body. And an example of flexion would be bending the elbow, and we know that flexion reduces the angle of a joint. And that concludes Chapter 7. Chapter 8. Intercalated discs, their location and function. We can see this on page 152. Intercalated discs will be located within cardiac muscle fibers. They allow the plasma membranes of adjacent cardiac fibers to contact one another. These are dark staining bands. They are cross-sections of intercalated discs that appear within the cross striations of cardiac muscle fibers. Key terms to know: sarcomere, actin, myosin, Z-lines, motor unit, fascia, and fascicles. Let's start with sarcomere. You can see that on the bottom of page 153. We know that the sarcomere is the basic functional or contractile unit of skeletal muscle. We understand the contraction of a muscle causes two types of myofilaments to slide towards each other, shortening the sarcomere and therefore the entire muscle. But when a muscle is relaxed, sarcomeres remain at their resting length and the filaments maintain their resting positions. Actin. Actin is also found on page 153. Actin is what our thin myofilaments are mainly composed of. It is a protein. So our thin myofilaments are composed mainly of the protein actin. Myosin is what composes our thick myofilaments. It is a protein that forms our thick myofilaments. Z-lines can be seen on page 154. We know that repeating units or sarcomeres are separated from each other by dark bands known as Z-lines or Z-discs. So Z-lines or Z-discs occur at the end of each sarcomere. Motor unit. We can see motor unit on page 158. A single motor neuron with the muscle fibers it innervates is known as a motor unit. Fascia and fascicles. We can see those on page 152. We know that fibrous connective tissue will wrap around each individual muscle fiber and continue as it wraps around groups of muscle fibers known as fascicles. So groups of muscle fibers are fascicles. They then form a wrapper around the entire muscle organ. Fascia is the loose connective tissue that is found outside the muscle organs, forming a flexible, sticky packing material between muscles, bones, and the skin. Know the difference between tonic contractions, isotonic contractions, isometric contractions, twitch contractions, tetanic contractions, and eccentric contractions. So we can see those on page 158 and 159. So we'll start with tonic contractions. Tonic contractions are going to be isotonic or isometric contractions. So we have isotonic contractions that produce movement of a joint. With this muscle of contraction, the muscle changes length and the insertion end moves relative to the point of origin. There are two types of isotonic contractions. One is concentric contraction, in which the muscle shortens, and the other is an eccentric contraction, in which the muscle lengthens, still providing work. And tonic contractions we're actually going to find on page 155. We know that a tonic contraction will maintain the muscle tone or posture of our body. So tonic contractions do not actually move body parts. They instead hold our muscles in position. Muscle tone maintains good posture, and this is accomplished with a tonic contraction. Whereas isotonic contractions actually produce movement at a joint. This would be your concentric contractions and your eccentric contractions. Twitch contractions can be seen on page 158. We know that it's a quick, jerky response to a stimulus that can be seen during isolated muscles during research, but we know that they play a minimal role in our normal muscle activity. Tetanic contractions are more sustained and steady. These are produced by a series of stimuli bombarding the muscle in rapid succession. These contractions will blend together, producing a sustained contraction or tetanus. Finally, isometric contractions do not produce any movement. Isometric contractions increase the muscle length. Isometric contractions don't increase the muscle length, they occur when the muscle contracts and no movement results. So therefore, during an isometric contraction, the length and during contraction and relaxation is about equal. You can see this on page 159. We know that isometric contractions will increase the tension within a muscle, but don't actually change the length of the muscle. It occurs when the muscle contracts, but no movement results. Muscles do not shorten, instead the tension within them increases. What does endurance training lead to? So we can see this on the bottom of page 159. Endurance training will not lead to muscle hypertrophy, but instead increases a muscle's ability to sustain moderate exercise over a longer period of time. This could be exampled by aerobic activities like running, bicycling, or other primarily isotonic movements, which allows the number of blood vessels in a muscle to increase without significantly increasing its size. The increased amount of blood flow allows a more efficient delivery of oxygen and glucose to muscle fibers during exercise. Aerobic training will also lead to an increase in the number of mitochondria within muscle fibers, allowing production of more ATP, which will be needed to fuel prolonged activities. What does strength training lead to? We can also see that on page 159. Strength training leads to muscle hypertrophy. This can usually happen by contracting your muscles against heavy resistance. Isometric exercises and weightlifting are common strength training activities. This type of training will result in an increased number of myofilaments within each muscle fiber. Although the number of muscle fibers will stay the same, the increased number of myofilaments will greatly increase the mass of the muscle. What major muscles move the following areas? The trunk, head and neck, upper extremities, and lower extremities. So let's look at our muscles of the head and neck. So we know that the zygomaticus elevates the corners of the mouth and lips and is known as the smiling muscle. We can see that our sternocleidomastoid and trapezius muscles work together to flex the head onto the chest. As far as our upper extremities, we know that the pectoralis major muscle covers the upper chest, and the latissimus dorsi muscle originates from structures over the lower back. Our pectoralis major is a flexor, and the latissimus dorsi an extensor of the arm. Our deltoid muscle is a powerful abductor of the arm. The triceps brachii is an extensor of the elbow, performing a straightening function. The rectus abdominis is one of the sheet-like muscles that appears as a band or strap-shaped muscle running down the midline of the abdomen from the thorax to the pubis. It protects the abdominal viscera, but also flexes our vertebral column. We know that our intercostal muscles and the diaphragm allow our chest to change size and shape during breathing, allowing air to move into or out of the lungs. We can see that our iliopsoas muscle is classified as a flexor of the thigh and also is an important postural muscle, stabilizing the trunk, keeping you from falling backwards when you stand. The tibialis anterior muscle dorsiflexes the foot. The gastrocnemius is our primary calf muscle, and it's responsible for plantar flexion of the foot. The muscles of our upper extremities are the pectoralis major, the latissimus dorsi, the deltoid, the biceps brachii, and the triceps brachii. The muscles of our trunk are the external and internal obliques, as well as the transversus abdominis. We also have the rectus abdominis, the intercostal muscles, and the diaphragm. Dorsiflexion versus plantar flexion. Where is it occurring? We can see this on page 160. Dorsiflexion and plantar flexion refer to ankle movements. In dorsiflexion, the dorsum or top of the foot is elevated with the toes pointing upward. In plantar flexion, the bottom of the foot is directed downward, as when you are standing on your toes. Key terms to know are flexion, extension, rotation, circumduction, abduction, and adduction. Understand the definition and how to use them in an example. We can start on page 159. We know that flexion is a movement that decreases the angle between two bones at their joint. It is commonly described as a bending movement, and an example is bending your elbow or knee. Extension movements are the opposite of flexion. They increase the angle between two bones at their joint. Therefore, it is a straightening or stretching movement rather than a bending movement. An example would be flexion and extension of the knee. Abduction means moving a part away from the midline of the body. An example is moving your arm out to your side. Adduction refers to moving a part towards the midline, such as bringing your arms down to your sides after they have been lifted. Rotation is a movement around a longitudinal axis. An example would be rotating your head and neck by moving your skull from side to side. Example would be shaking your head no. Circumduction moves a part so that its distal end moves within a circle. A great example is when a pitcher winds up to throw a ball, they will circumduct their arm. So those would be examples and definitions of those movements. You can also see pictures of these movements on page 160 and 161. Which body systems make movement possible? We can see this on page 156. We know that our skeletal muscles cause movements by pulling on bones across movable joints, so our skeletal system interacts with the muscular system in order to produce movement. We also know that our respiratory, circulatory, nervous, muscular, and skeletal systems all play essential roles in producing normal movements. And this is on page 156 at the very bottom. How much does skeletal muscle weigh? Well, if we flip to the first page of the chapter on page 151, we can see that in a 120-pound person, about 50 pounds of them is skeletal muscle. Striated muscles and striations. Where are they found, and is there another name for striated muscle? We can see this on page 152. We know that another name of skeletal muscle is striated muscles due to its cross stripes or striations. Those are characteristic of striated muscle. Skeletal muscle is also known as voluntary muscle. We also know that striations are found within cardiac muscle fibers. They have cross striations. They also contain the intercalated discs. Smooth muscle contains no striations. Therefore, they are sometimes called non-striated muscle fibers. It's important to understand that smooth muscle fibers are involuntary because we normally have no control over their contractions. Because of their location in many hollow internal organs, as well as the walls of blood vessels, we can also refer to smooth muscle as visceral muscle. Does the muscle insert onto the more stationary or mobile part of a bone? Well, we can see this on page 153. We know that the muscle's attachment to the more movable bone is known as the muscle's insertion. The muscle's attachment to the more stationary bone is called its origin. We know that the contraction process of a muscle cell requires energy, and most oftentimes we get this energy from ATP. Remember this simple rule, which you can see on page 155. A muscle's insertion bone moves towards its origin bone. And that concludes chapter 8. Moving on to chapter 16. Chapter 16, chyme, definition, purpose, and location. We can see this on page 369. We know that our muscle fibers are able to break up chunks of ingested food into tiny particles and mix them thoroughly with gastric juices, forming a semi-solid mixture known as chyme. Where we can find chyme is within the stomach, propelling it down the digestive tract. Alimentary canal versus the GI tract. Is there a difference? No, there is not. It is the same exact thing. And you can see this on page 361. Our long winding tube of our digestive system is the alimentary canal, and the same term can be used, which is the gastrointestinal tract. The main organs of our digestive system, what are they? As well as the accessory organs of our digestive system, what are they? We can see this on page On page 361, our main organs of digestion are those making up the alimentary canal, and the accessory organs of digestion are not segments of the tube, but instead are within or surrounding that tube. You can see a great breakdown of this on page 362 within table 16-1. Digestion, where does it occur? Which organs break down which substance? What breaks down lipids? What breaks down proteins? What are the end products of digestion of these substances? For example, what is the product of protein digestion? So we know that digestion, and we can see on page 362, occurs within the entire GI tract. So complex foods need to be taken into the GI tract when we ingest them. Then our ingested material is broken down into simpler nutrients via the process of digestion. This breakdown or digestion of food is mechanical and chemical in nature. It occurs throughout our digestive tract or throughout our GI system. The teeth will be used to physically break down large chunks of food before it's swallowed. The churning of food in the stomach will then continue the mechanical digestive process. To break down larger chunks of food into smaller bits and move it along the GI tract, we have movement or motility of the GI wall. In chemical digestion, large nutrient molecules are reduced to smaller molecules. This process requires secretion of digestive enzymes and other products into the lumen of the GI tract. After the digestive processes have altered the chemical and physical composition of ingested food, the resulting nutrients are ready for the process of absorption. Absorption is the movement through the GI mucosa into the internal environment. So let's look at what breaks down each of our nutrients. And we can see this on page 378. So we know that very little digestion of carbohydrates occurs prior to reaching the small intestine. After our carbohydrates reach the small intestine, pancreatic and intestinal juice enzymes will digest the starches and double sugars. A pancreatic enzyme known as pancreatic amylase starts this process by breaking down polysaccharides like starches into disaccharides. Three intestinal enzymes known as maltase, sucrase, and lactase will digest these disaccharides, changing them into monosaccharides or simple sugars. The end product of carbohydrate digestion are monosaccharides, and the most abundant of which is glucose. Protein digestion begins in the stomach. Hydrochloric acid and gastric juices will help unfold large complex protein shapes so that digestive enzymes can reach these peptide bonds holding the amino acids together. The end product of protein digestion is amino acids. Just as with carbohydrates, very little fat and oil digestion occurs prior to reaching the small intestine. So lipid digestion occurs in the small intestine. The end products of lipid digestion are triglycerides that are broken down into fatty acids and glycerol. So the end products of lipid digestion are fatty acids and glycerol, and lipid digestion begins in the small intestine. We know that our salivary glands and the pancreas secrete a digestive enzyme known as salivary amylase, as well as pancreatic amylase. This begins the process of breaking down complex carbohydrates. Chemical digestion requires enzymes for this to happen. You can see more about this on page 378. We know that enzymes are protein molecules acting as catalysts, and during chemical digestion, certain enzymes will very selectively speed up the breakdown of specific nutrient molecules, but no others. Enzymes responsible for speeding up the breakdown of lipids, for example, will have no effect on carbohydrates or proteins. And that is the function of enzymes. Anatomy of the common bile duct. What does it do and what does it connect to? We can see this on page 373. So we can see that the cystic duct and the hepatic duct join to form the common bile duct. Our cystic duct leads to the gallbladder. You can also see a great image of this on page 372. But we can see that the cystic duct and the hepatic duct will join to form the common bile duct. Very easy to see this in the green portion of the diagram on page 372. And what does our common bile duct do? It drains bile into the small intestine, specifically the duodenum. Key terms: digestion, absorption, mechanical digestion, and chemical digestion. So we've already pretty much covered all of those. Chemical digestion can be read more about on page 378. You can see the broad definitions of these terms on page 362. Where we can see that the mechanical process of breaking down food is mechanical digestion, and then chemical is using the enzymes. converting the large nutrient molecules to smaller nutrient molecules. We know that absorption is the movement through the GI mucosa to the internal environment. Where does mechanical digestion occur? What organs accomplish this? Are enzymes or secreted substances that aid in this process? So we know that the mouth and the teeth begin the process of mechanical digestion. It then continues within the stomach. We know that the oropharynx on page 368 is most actively and directly involved in our digestive process because it plays an important role in a coordinated type of GI tract motility involved in swallowing or deglutition. Then we're going to have our pyloric sphincter, which is going to help move It's going to help move chyme that is held in the stomach into the small intestine. So the pyloric sphincter, which you can see on page 370, takes food from the stomach and moves it into the small intestine. It allows for partial digestion of proteins after the chyme has been held within the stomach. After chyme has been mixed within the stomach and protein digestion begins to get underway, it starts its passage through the pyloric sphincter and into the first part of the small intestine. Bile, function, importance, and where it comes from. So we can see this on page 373. Liver cells secrete a substance known as bile into ducts, which is why the liver is classified as an exocrine gland. It is the largest gland in the body. Exocrine gland in the body. Bile contains a mixture of substances. Some of these have direct digestive functions. The liver also serves an excretory role because it removes the yellowish-brown bile pigments that are formed from the breakdown of hemoglobin from old red blood cells and puts them into the bile for elimination from the body. Our liver continuously secretes bile. Our hepatic ducts specifically drain bile out of the liver. We know that chemically, bile contains significant quantities of cholesterol and substances known as bile salts that will act as detergents to mechanically break up or emulsify lipids. And that's on page 373. The length of the GI tract, we can see on page 361. In an adult, our hollow tube of the GI tract is about 9 meters or 29 feet long. The number of permanent teeth in an adult. We can see this on page 366. By the time a young adult is somewhere between 17 and 24 years old, a full set of 32 permanent teeth is generally present. 32 permanent teeth. Pyloric sphincter we already talked about. Serosa, function, importance, and definition. So we can flip back onto page 364. The serosa is the outermost covering or coat of the digestive tube. It is composed of serous membrane. It can also be known in the abdominal cavity as our visceral peritoneum. It will double fold of serosa extending between the visceral and parietal layers not only helps anchor the organs of the digestive tract to the posterior abdominal wall, but it also helps keep these abdominal organs in their places. It is the outermost covering or coat of the digestive tube. The submucosa can be seen on page 364. It is a connective tissue layer lying just below the mucosa, and it contains many blood vessels and nerves. It's important to note the layers of the wall structure of the digestive tract, noting that from the inside coat to the outside of the tube, we have the mucosa or mucous membrane, the submucosa, the muscularis, and the serosa, which can all be seen and read about on page 363. Dentin. So we can see dentin, where it is found and its purpose, on page 366. The crown is the portion of the tooth that is exposed and visible within the mouth. It is largely made of a bone-like material known as dentin. Dentin is then covered by enamel, our hardest tissue within the body. Enamel is ideally suited to withstand the grinding that occurs during the chewing of hard and brittle foods. And amylase we already talked about. It's important to also note on page 371 that most of the chemical digestion occurs in the first subdivision of the small intestine, known as our duodenum. Part of what makes our small intestine so adept at performing digestion and absorption are the several structure features of the lining of the small intestine. These would be plicae, the multiple circular folds, and villi, which cover these folds as thousands of tiny little fingers. They help to increase the surface area, providing a large absorptive surface area, allowing for a faster absorption of nutrients from the intestine into the blood and the lymph. And that concludes the review of the study guide for Structure and Function Exam 2.